So good evening, students. So welcome to another session. So in the previous session, we started discussing about a few classification with regards to mutation, right? So under the classification, we started off with something called as the chromosomal basis of classification, right? Chromosomal aberrations, right? Broadly, if you could remember, just to recollect what we have seen yesterday, we started classifying all the mutations into two categories based on where the mutation is happening, whether it is happening on the chromosome or whether it is happening on the gene. So if there is mutation happening on the chromosome, we call it as chromosomal mutation. And if there is mutation happening on the gene, we started calling it as gene mutation. So with regards to chromosomal mutations is what we took up in the previous session, right? So we said that these mutations in the chromosome could lead to two different things. One is there could either be some sort of changes in the structure of chromosome, or there could be some sort of changes in the number of chromosomes. So if there is changes in the structure of chromosome, we started calling it as chromosomal aberrations, or we also call it as morphological mutations, or we also call it as structural mutation. If there is change in the number of chromosome, we started calling it as numeric mutations or genomic mutation. Under the first category, which is chromosomal mutation, under that, the first category what we saw was structural mutation. And under structural mutation, again, we saw four different classes of mutations. One was said to be deletion, second one was duplication, third one was inversion, and the fourth one was translocation. And under translocation, we saw again two different types of translocation. One was called interstitial translocation, and the second one was called reciprocal translocation. So we have seen what exactly is happening in each of these cases. In the next segment, what we saw was we took up with numeric we took up with numeric mutations, or we also call it as genomic mutation. So under this numeric mutations or genomic mutations, we saw two different categories again. One was called euploidy and the other one was called aneuploidy. So in case of euploidy, we said that there is change in the complete set of chromosomes. So haploid, diploid, triploid, tetraploid, polyploid, and those are the things what we saw. With regards to the second type again, that is aneuploidy, or decrease in either one chromosome or two chromosomes. So decrease in one chromosome, we call it as monosomy. Decrease in two chromosome, we call it as nullisomy. Increase in one chromosome, we call it as trisomy. Increase in two chromosomes, we call it as tetrasomy. And we also saw that the reason for this aneuploidy is mostly a process called as non-disjunction, which is failure of the separation of chromosomes during the anaphase stage. So the result of this non-disjunction would lead to this aneuploidy. So that's where we have stopped in the previous session. So we started off with the chromosomal mutations and we have done with chromosomal mutation. So in this session, let us take up with gene mutation and we'll see a few things with pertaining to gene mutations. So let's take up with gene mutation. So under gene mutations, if you could remember, we said if there is some sort of changes happening in the gene, that type of mutation, we call it as gene mutation. So if there is changes happening in the chromosome that we saw in the previous session, we called it as chromosomal mutation. And here we are talking about the mutations that occur in the gene, within the gene of a particular chromosome. Such type of mutations, we start calling it as gene mutation. So let us take up with regards to this gene mutation. So we said that gene mutations could be classified again into three different categories, right? Gene mutation would be classified into three different categories, of which the first one, we call it as inversion. The first one was inversion, what we saw. The second one was substitution reaction or substitution mutation. Substitution mutation, or we also call it as replacement mutation, or we also gave one more term also, we also called it as point mutation in the previous session, right? So this is the second one. And the third one what we have is called the frame shift mutation. So let us see in detail what exactly are these three types of mutation. Let us first begin with inversion. Inversion is actually similar to what we have actually seen even in case of chromosomal mutation, right? So even in case of chromosomal mutation also we saw inversion. So with regards to that, what did we see? If this is a chromosome here, right, 1 2 3 4 5 6 7 8 and 9, what did we say? There is inversion happening. So these two genes, gene number 6 and gene number 8, they are flipping over to 180 degrees so that they are getting inverted in their position. That we call it as inversion with regards to chromosomal mutation. Here also, we'll be talking about the same type of inversion again. Let us consider a triplet codon. Say we have GAG. I think all are familiar with this, right? Triplet codon. So this is one codon here, triplet codon, which codes for a particular amino acid, right? Now, what exactly is happening is there is some sort of changes happening in this particular triplet codon. So we are talking about that change. That first change is nothing but inversion. So what exactly is happening in inversion is, say we have this A here at the center. So that A will come to the position where the second G is being there, and this G will again come to this particular position. So initially the codon was GAG, but after the mutation, which we call it as inversion type of gene mutation, right? The codon now has been changed to G, G, and A. This is how it is changed. This sort of mutation, we start calling it as inversion mutation. GAG was producing, say, amino acid number 1, right? But once GGA is formed, this amino acid 1 will no longer be produced, right? So that is what we start calling it as inversion type of gene mutation. This we call it as inversion type of chromosomal mutation, and here this would be inversion type of gene mutation, where the two codons, I mean the two nucleotides will actually, or the nitrogen bases would get inverted in their positions. So they flip of over 180 degrees, right? So there is change in their positions happening. So this type of mutation, we start calling it as inversion type of gene mutation. Now let's talk about the second one, which is called as substitution mutation, or it is also called as replacement mutation, or we also call it as point mutation. So this is also called as point mutation. Now what does this point mutation mean? At only one particular point, there is change in the nitrogen base happening, right? So we have a gene. In that gene, we have a particular codon. In that particular codon, again, there is only one nitrogen base change is happening. So if that is the condition, we start calling it as point mutation, or we also call it as substitution mutation, or we also call it as replacement mutation. Let us take up this example what we have already seen in the previous classes, which is sickle cell anemia. I hope all of you remember this sickle cell anemia. So if you could recollect, we saw about the sickle cell anemia while we were discussing about a topic called as pleiotropy, right? So while discussing about pleiotropy, we came up with this particular disorder called as sickle cell anemia. So what happens in the sickle cell anemia? The shape of the RBC changes, right? Changes from a biconcave structure or a saucer shaped structure to a sickle shape, right? So it is actually a saucer shaped RBC. So this RBC would change into a sickle shaped structure. So this would be the RBC, right? So if RBCs are present like this we start calling it as sickle cell RBC, and that disorder we actually call it as sickle cell anemia. Now, what is the reason for this particular change? If you could recollect what we have seen while we were discussing about pleiotropy, we said that we know that hemoglobin is a tetrapolymer. So there are 4 polypeptide chains present in case of hemoglobin. We know that there are 2 alpha chains as well as 2 beta chains. So I told you that while discussing about pleiotropy there, a mutation in one of the beta chains of hemoglobin, right? So there is a mutation happening in the beta chain of the hemoglobin. And what sort of mutation it is? It is nothing but a point mutation. So let's pick up with the beta chain. Just imagine that this is the beta chain of the hemoglobin. Beta chain of the hemoglobin. So there are a lot of amino acids which constitute the beta chain of amino acid. So here there is one amino acid, 1, 2, 3, 4, 5, 6, 7, and it goes on like this to form a long chain of peptide. So this would be 1, 2, 3, 4, 5, 6, 7, and it goes on like that. So we are actually interested in the 6th amino acid. So we know that 6th amino acid, it codes for something called as glutamic acid. right? So the sixth amino acid of the beta polypeptide chain of hemoglobin actually codes for amino acid, right? So if you see the codon for this, so the codon here is actually GAG. So GAG actually codes for this particular amino acid called as glutamic acid. But due to this point mutation, glutamic acid is not being coded. Rather than that, there is one more amino acid which is actually being formed. So where exactly is this mutation happening? The mutation is actually happening here in this particular A of that triplet codon, where A is replaced by U. Is being replaced by U. As a result of which, what exactly is happening? Rather than glutamic acid which is being formed, in place of glutamic acid, valine is actually formed. Right. So this we start calling it as point mutation. So why exactly it is called point mutation? Because there is only one there is only one nitrogen base change happening in that particular triplet codon. So since there is only one amino acid change or one nitrogen base change happening, such type of mutation, we call it as point mutation. Now, what did we say? We also gave another term called as substitution mutation. So what exactly is happening? U is being substituted by A, right? Again, we also gave one more term also. We also call it as a replacement mutation, where U is replacing A. As a result of which, the amino acid, instead of glutamic acid, valine is actually being formed. Now, since valine is formed in the sixth position of the beta polypeptide chain, the structure of hemoglobin will be altered. Now, since the structure of hemoglobin is altered, the structure of RBC also would be changed, right? So one single nitrogen base change would lead to this sort of a disorder, which is called as sickle cell anemia. And the reason for that is this point mutation or the substitution mutation, or it is also called as replacement mutation, right? So what exactly is happening in case of inversion? In inversion, we said that if there is GAG, the positions are being changed, as a result of which this is being changed to GG and A. So instead of GAG, it is being changed to GG and A. That is with regards to inversion, the first one. Talking about the second one, substitution or point mutation, only one nitrogen base is being changed, right? And that nitrogen base, one best example what we can take is this sickle cell anemia, where in the sixth amino acid position of the beta chain of hemoglobin, instead of glutamic acid, valine is actually being expressed. The reason for that is GAG is being changed to, or it is being mutated to GUG. So what exactly is getting mutated? A is being mutated, as a result of which U replaces A. So therefore, we start calling it as point mutation, because there is only one single nitrogen base change happening. As a result of this, what would happen? Instead of glutamic acid, valine is being expressed. Since valine is being expressed, what would happen? The hemoglobin structure would be altered. Now, since the hemoglobin structure gets altered, the shape of the RBC also, it becomes a sickle shape. Therefore, we start calling it a sickle cell anemia, right? So this is what we have with the second type of gene mutation, which is called substitution mutation or point mutation. Now, this substitution mutation or point mutation could be again of two different types, right? It could be of two different types. One, we start calling it as transition type of point mutation. The second one is called transversion type of point mutation. So the first one is called transition type of point mutation. The second one is called transversion type of point mutation. Now, what is this transition type of point mutation? If one purine is replaced by another purine, right? Just like what we said in point mutation, what you need to remember, there is only one nitrogen base change happening. So one nitrogen base is being replaced. So if a purine is replaced by another purine, such type of point mutation, we call it as transition type of point mutation. There is one more possibility also. If a pyrimidine is replaced by another pyramidal. Right? So if a purine is replaced by another purine, we also call it as transition type of point mutation. If a pyrimidine is replaced by another pyrimidine, that type of mutation also we start calling it as transition type of point mutation. Now, what could be transversion type of point mutation? Opposite of this. If a purine is replaced by a pyrimidine, right? If a purine is replaced by a pyrimidine, then we call it as transversion type of point mutation. It could be the other way around also, where a pyrimidine would be replaced by a purine. So the other way around also, we call it as transversion. So simple things, what you need to remember here with regards to substitution mutation or point mutation, the mutation is happening with corresponds to only one nitrogen base, right? So there is only one nitrogen base change happening. So if a purine is replaced by another purine, or if a pyrimidine is replaced by another pyrimidine, such type of mutation we call it as transition type of point mutation. Whereas if a purine is replaced by a pyrimidine, or a pyrimidine is replaced by a purine, such type of mutation we start calling it as transversion type of point mutation. Right? So this is the second type of gene mutation what we have, which is called as point mutation or substitution mutation. Now let's take up with the third one, which is called the frame shift mutation. Right, so let us take up with the third type, which is called the frame shift mutation. So talking about frame shift mutation, so let me give you a simple idea of what exactly this frame shift mutation is all about. Frame shift mutation. So what exactly does this frame shift mutation mean? So to understand what exactly is this frame shift mutation, let us take up a simple sentence here, so that you get a clear picture of what exactly frame shift mutation is. So the sentence here what we have is, why did you eat the egg? So let us take up this as a simple expression here, a simple sentence here. Why did you eat that? The reason why I chose this again, because all the letters have only three words. Sorry, each word has only three letters, right? These three letters would start indicating each triplet codon. We know that each triplet codon would code for one amino acid. So consider this to be one triplet codon. This is one triplet codon. So all these to be one triplet codon. Right. So this one would synthesize one amino acid, second one another amino acid, this one more amino acid, this one more amino acid, this one more amino acid, and the last one another amino acid. Right. Now each one we start calling it as a frame. Right. Try to understand this. Each of the triplet codon is actually referred to as a frame. Right. So we have 1, 2, 3, 4, 5, and 6 frames in this. Now we are talking about a mutation, which is called as frame shift mutation. Now, before we get into this, frame shift mutation could be of two types. One is, we call it as insertion type of frame shift mutation. And the other one is said to be deletion type of frame shift mutation. One is said to be insertion and the other one is said to be deletion. Let us first begin with insertion type of frame shift mutation. So what does insertion mean? Insertion is nothing but we are inserting something. Now just imagine that in this place between Y and Z, we are actually inserting, say, some X here. So X is being inserted in this particular place. Now what happens if X is inserted in this place, right? So the first one would be as usual Y. The second one, if you see here, rather than Z, what would be the second triplet codon here? It would be XID. It would be X, D, and I. X, D, and I. So this would be the second frame here. The third frame would be with this D, U, O. The fourth frame would be Y, E, A. The next frame would be T, T, H. Then another frame would be E, E, G, and this one would be G here. So if you start observing this, what exactly is happening? The frames are actually being changed here. The first frame is as usual it is, but the second frame should be DID. So what exactly is happening? It has become XDI. The third frame should be YOU, but what has happened here? It had become DYO. The next frame has to be EAT, but what has happened here? UEA. So like this, in all of the cases, the frame has been shifted. As a result of which, say amino acid 1 is synthesized here, 2, 3, 4, 5, and 6. But here only amino acid 1 would be synthesized, whereas all the others would start synthesizing some other amino acids. As a result of which, what would happen? The proper functioning of the body would be completely altered. So this we start calling it as frame shift. Why do we call it as shift? Because the frame is being shifted, right? So this is one frame. The first frame is unaffected, but due to insertion of one nitrogenous base, what exactly is happening? The frame has been shifted towards the right. So this is very important what you need to remember. When a nitrogen base is added, the frame always shifts towards the right. So insertion always leads to the shifting of the frame towards the right. So the frame starts shifting towards the right. So this is the first type of frame shift mutation what we have, which we call it as insertion type of frame shift. Right. So this is what we have. Now, let us get into the second type, which is deletion type of frame shift. So this is with insertion. So this would be insertion because of insertion of this one here. Now let us talk about the second condition there, which is nothing but deletion. What happens in deletion? In deletion, there is deletion of one nitrogenous base happen. Now just let us imagine that this D here is being deleted. From the second frame, D is being deleted. Now if D is deleted from the second frame, what would be the condition now? First frame unaffected. Why? What would be the second frame now? D is not there. So what would happen? It would be I, D, and U. So second frame would be I, D, U. Third frame would be O, U, E, right? The next frame would be A, P, and T, A, T, T. The next frame would be H, E, and E, right? The last frame would be G and G. So what exactly is happening here again? The first one is unaffected, but again, the frames are being shifted. This we start calling it as frame shift mutation. So due to deletion of one of the nitrogenous base, so what exactly is happening? The frame is actually getting shifted. So here what exactly is happening? The frame is actually shifted towards the left. So in case of deletion, the frame shifts towards the left, whereas in case of addition, the frame starts shifting towards the right, whereas in case of deletion, it starts shifting towards the left. So this is what we start calling it as frame shift type of mutation. So this would be with regards to deletion. Right. So this is again another type of mutation, what we call it as frame shift mutation, which is actually seen only in case of genes but not in the chromosome, right? So these are the three different types of gene mutations what we have, which is called as inversion, substitution mutation, as well as the frame shift mutation. In inversion, what is happening? There is inversion of the nitrogenous base happening within a triplet codon. In substitution, what is happening? A particular base is being substituted by another base. It could be either purine getting substituted by a purine, or it would be pyrimidine getting substituted by a pyrimidine. If that is the case, we started calling it as transition type of point mutation. Second one category what we saw was transversion type of point mutation, where we said that either a purine is replaced by a pyrimidine or a pyrimidine is being replaced by a purine, right? So that we started calling it as transversion type of point mutation. The third type of mutation what we have is called the frame shift mutation, which could be either insertion frame shift or deletion frame shift. In insertion, what exactly is happening? The frame is actually being shifted there because of the addition of one of the nitrogen bases. So the frame is being shifted. Whereas in case of deletion, what exactly is happening? Again, due to removal or deletion of one nitrogen base, again, the frame is being shifted back. So this we start calling it as deletion type of frame shift mutation, right? So these are the three different types of gene mutations what we have. So on a whole, if you have to classify mutations broadly, this is the major classification what we have with regards to mutations. So mutations could be broadly classified into two categories: chromosomal mutation, and the other one is called gene mutation. In chromosomal mutation, there is changes happening in the chromosome. In gene mutation, again, there are these changes that actually happen in this particular genes, right? So in the later sessions, we'll take up a few disorders pertaining to chromosomal disorder, chromosomal mutation, as well as certain disorders pertaining to the gene mutation also. So we'll get into the details of each of the disorders with regards to chromosomal mutation as well as gene mutation also. So this is the major classification what we have with regards to mutation. So along with this, there is one more classification of mutations also, right, where mutations could be classified again into two categories, right? So those we start calling it as somatic mutation, and the other one is called germinal mutations. So this is one more classification, somatic mutation, and the other one would be termed as germinal mutation, right? So this would be our third classification what we are seeing. So if you could remember, in the previous sessions we took about two classifications. The first classification what we saw was based on the origin of mutation. We started classifying mutations into two categories, that is either spontaneous mutation and the other one is induced mutation. Second category was based on where exactly mutation occurs. We again classified them into two categories: chromosomal mutation and gene mutation. So this would be our third classification of mutation, right? So mutations could be again classified into two categories based on which type of cells in the body mutations occur. So we have somatic mutations as well as the germinal mutation. So what are these somatic mutations? Mutations that occur in the vegetative cells of the body, or the body cells, we start calling it as somatic mutation, right? So mutations that actually occur in simple, we can say in any of the body cells or vegetative cells, those type of mutations we start calling it as somatic mutation. What about germinal mutation? Germinal mutation is usually seen in the germinal cells. Germinal cells are nothing but the cells which reproductive cells. Germinal cells or the reproductive cells. So either in the germinal cells or the reproductive cells, if there is mutation happening, such type of mutations we start calling them as germinal mutation. One more important thing what you need to remember here is somatic mutations are not inherited, right? Somatic mutations are not inherited. So one example, let me give you. Say suppose a person is suffering from skin cancer, right? Skin falls under the category of somatic cells, right? Skin cells, right? So it is a somatic mutation. So that skin cancer cannot be inherited to the progeny, right? So that type of mutations we start calling them as somatic mutations, which are actually not inherited. Whereas the mutations that occur in the germinal cells, these mutations are said to be inherited mutations, right? So these mutations are said to be inherited mutations. So somatic mutations, they are not inherited. Germinal mutations, they are actually said to be inherited, right? So this is what we have with complete things with regards to the topic called as mutation. So we saw what exactly mutation is all about. Then we saw what exactly causes mutation. Then we saw how do we classify mutation on different factors. So this is what we have with a small topic called as mutation, which was actually first put forth by a scientist by name Hugo de Vries while working on the plant called as Inothera lamarckiana, which is called as the evening prime rose, right? So with this will be done with mutation. Now let's get into another topic for today, right? So let's begin with another type of inheritance pattern. So we are still talking about inheritance. So let us see another type of inheritance. So the type of inheritance what we are going to talk about now is called as cytoplasmic inheritance, right? So we start calling it as cytoplasmic inheritance. So what exactly is this cytoplasmic inheritance? Till now we have been talking a lot about the inheritance, right? So till now we have been talking a lot about inheritance, right? And we saw that whenever we talk about inheritance, till now we have been talking about the inheritance of a particular character from the genomic DNA. So when we say genomic DNA, it is nothing but the DNA which is present in the nucleus, right? So we know that DNA is present in the nucleus, or rather we can put it this way: chromosomes are present in the nucleus, and these chromosomes are the ones which carry the genes, and genes are the ones which are responsible for passing on a character from one generation to the other generation. That's what we have been talking about till now. But now what we are going to talk about is something called a cytoplasmic inheritance. So what does the cytoplasmic inheritance mean? Along with nucleus, there are certain other organs also, so rather organelles also, which carry their own DNA. Try to understand this. You would have studied this in the previous year, right? So along with the nucleus, there are certain cellular organelles which carry their own DNA. So we start calling those organelles as what? We start calling them as semi-autonomous organelles. Semi-autonomous organelles, right? So we start calling them as semi-autonomous organelles. So along with nucleus, right, DNA is also present in certain organelles, which we start calling them as semi-autonomous organelles. So what are those organelles? There are two major organelles which have their own genetic material, which have their own DNA. One is mitochondria. So one is mitochondria, and the other one is plastid. So these two cellular organelles, we start calling them as semi-autonomic cellular organelles. Why do we use the term semi-autonomous here? If it is autonomous, it would be able to, when do we say an organelle is autonomous? When it has its own nuclear material, or when it has its own DNA, and it is able to control or govern its functions by itself. That time we use the term as autonomous organelles. Only one autonomous organelle we have, and that would be the nucleus, right? And here we are calling these two as semi-autonomous. Why do we call it as semi-autonomous? Reason number one, because they have they have DNA within their organelle. Since they have DNA, they would be autonomous, but we don't call them as fully autonomous because their work, they cannot do their work by themselves completely. They are dependent on the nucleus for carrying out some sort of work. That's the reason why we use the term here semi. Okay, so keeping that aside. So there are two semi-autonomous organelles present in the cell. Mitochondria is present in both plant cell as well as animal cell, whereas plastids is present only in case of plant cell. Right. Now these two organelles, since they have their own genetic material, all right, so in that genetic material also there are certain genes present, and those genes again would be transferred or inherited to the progeny, right? Try to understand this, right? Along with the nuclear genetic material, these organelles also, since they have their own DNA, now we know that since they have their own DNA, they also have their own genes, and those genes from these two organelles would be inherited to their progeny. Such type of inheritance we start calling as cytoplasmic inheritance. Getting it? Why do we call it as cytoplasmic inheritance? Because these two, where are these two organelles present? They are actually present in cytoplasm. They are actually present in cytoplasm. Since they are present in cytoplasm, we start calling it as cytoplasmic inheritance, right? So let me again go through what exactly is this cytoplasmic inheritance all about. Till now, what we have seen is that we have a nucleus, and in the nucleus we have the genetic material being present, and in that genetic material, which is nothing but DNA, or rather the chromosome, in the chromosome we have the genes being present, and those genes we know are the ones which are responsible for the expression of a particular character, right? Now once the character is being expressed, as the genes are passed on to the progeny, right, as the genes are passed on to the progeny, right, so those characters are actually being inherited. That is what we have seen about till now about the inheritance. The same inheritance was actually given by Mendel. The same inheritance was given by Morgan also. So till now, what we have seen about inheritance pattern was all coming from the nuclear genetic material. But Along with the genetic material that is present in the nucleus, there are certain cellular organelles also which have their own genetic material, which is nothing but DNA. So which are those organelles? Mitochondria and chloroplast. Along with the nuclear DNA, mitochondria and plastids also have their own genetic material. Right. Since they have their own genetic material, and since they are also under the control of the nucleus, we start calling them as semi-autonomous organs. Right. Now, since mitochondria and plastids also contain DNA, their DNA also has genes, and those genes are also passed on into the offsprings, right, during the process of sexual reproduction. So the characters which are present in the genetic material of the mitochondria and plastids are also passed on into the progeny. They are also inherited into the progeny. Now, since these two organelles are present in the cytoplasm, we start calling such type of inheritance as cytoplasmic inheritance. This is the background of cytoplasmic inheritance, what we have. Right. Now, when we talk about cytoplasmic inheritance, let's get to the basics of the process of reproduction. I hope all are familiar with the process of reproduction, especially during the process of fertilization, if you see, right? So we have the female gamete here. So this is the female gamete. So we have the germinal cells. A diploid germinal cells will undergo the process of gametogenesis to form a haploid gametes. So here we have the female gamete. This female gamete, it has the nucleus, right? And along with that, there is this cytoplasm also present here. So we would have already studied that during the process of sexual reproduction or during the process of fertilization, the female gamete contributes the nucleus as well as the cytoplasm, whereas the male gamete, right, if you identify the male gamete here, which is the sperm, this male gamete will give only the nucleus, right? So during the process of fertilization, it is the female gamete which contributes the nucleus as well as cytoplasm, whereas the male gamete would contribute only the nucleus. Because if you could remember, when we were talking about the process of spermatogenesis, right, the last phase of spermatogenesis, where we said that the spermatids would be transformed into sperms, and during that particular process we saw about some changes that happens in the spermatid where they would be transformed into sperm. One of the changes what we saw, if you could recollect, we said that the cytoplasm would be completely reduced and some of the cytoplasm would be seen only in the tail region. So cytoplasm is completely reduced in case of the sperm, whereas the cytoplasm is present in case of the eggs. Now, when these two combine together in the offspring or in the zygote that is actually formed here, the zygote will have nucleus of the mother, that is nothing but the genetic material of the mother, the genetic material of the father, as well as the cytoplasm of the mother. So just let us imagine that we are talking about humans. In case of humans, this is the condition here. And we have already seen that in the cytoplasm, there is the cellular organelle called mitochondria present. So whatever genes are present in the mitochondria, those genes which are present in the mother, cytoplasm of the mother, would be seen present in the cytoplasm of this zygote also. So what exactly is happening? The genetic material, right? So the zygote is having genetic material from the mother, nuclear genetic material from the mother, nuclear genetic material from the father, and at the same time mitochondrial genome or mitochondrial genetic material from the mother also. So this type of inheritance pattern we start calling it as cytoplasmic inheritance. So there is one more name also given for cytoplasmic inheritance. It is also called as maternal inheritance. Maternal inheritance. Now what does this mean, maternal inheritance? Maternal is something to do with the mother, right? So the type of inheritance which is seen from the mother, we start calling it as maternal inheritance, because it is the mother who is contributing the cytoplasm during the process of fertilization. So whatever cytoplasm is present in the egg of the mother, the same cytoplasm would be present in the zygote also. So this type of inheritance, we start calling it as maternal inheritance or cytoplasmic inheritance. There is one more name also given for it. We also call it as organal inheritance. Organal inheritance. So why again do we call it as organal inheritance? Because it is the organelles which are present in the cytoplasm which are getting inherited into the progeny, right? So we call it as cytoplasmic inheritance or maternal inheritance, or we also call it as organal inheritance, right? So this is what we have with a brief introduction with regards to cytoplasmic inheritance. So with regards to this, we'll be taking up two examples. One will talk about a plant example, and the other one will take up with some animals also, right? So we'll be taking up with two examples. One is plants. We'll see how exactly the cytoplasmic inheritance occurs in case of plants. So for that, we'll be choosing a plant which is called as Mirabilis jarapa, and the other one will be talking about the snails, right? So cytoplasmic inheritance in case of snails, right? So with regards to the coiling of the snails, right? So this is the basis of chromosomal or rather cytoplasmic inheritance. So what we have discussed today is with regards to the process called as mutation. So under that, we spoke about something called as the gene mutation. So we have done with the gene mutation. So we saw three different types of gene mutations. One was called as inversion type of gene mutation. Second one was substitution mutation, and the third one was the frame shift mutation, right? So we went into details about all of these three mutations. So I hope there is no problem with all of those mutation types. Then we started off with cytoplasmic inheritance. So what exactly is cytoplasmic inheritance? Along with the nuclear material or along with the nuclear genetic material, certain cellular organelles also have, right? Certain cellular organelles also have their own genetic material. So two cellular organelles are present which have their own genetic material. One is called mitochondria as well as the plastid. So since they have their own genetic material, we start calling them as semi-autonomous organelles. Right. Now, since they have their own genetic material, and since these two organelles are present in the cytoplasm, right, during the process of gamete formation and during the process of fertilization, since the cytoplasm, since the mitochondria and plastids both are present in the cytoplasm, right? So during the process of fertilization, when the female gamete fuses with the male gamete, in the offspring, in the progeny will have nuclear material of the mother, nuclear material of the father, as well as the mitochondrial material also, or organelle genetic material also in the zygote. So we have already seen that during the process of fertilization, it is the female gamete who contributes the nucleus as well as cytoplasm, whereas the male gametes contribute only the nucleus. As a result of which, whatever cellular organelles are present in the cytoplasm, so those genetic material of those cellular organelles would be present in the zygote also. So say suppose a gene A is present in the mitochondrial genome. Now the same gene A from the mitochondrial genome, if it is present in the mother, the same gene A would be actually seen even in the child also, even in the offspring also. So such type of inheritance, we start calling it as cytoplasmic inheritance, because the cellular organelles present in the cytoplasm are the ones which are actually being inherited. The same type of inheritance, we also call it as maternal inheritance, because whatever genetic material is present in the mother, right, in the cellular organelles of the mother, so those genetic material is being transferred to the offspring through the mother. So therefore, we also call it as maternal inheritance, and we also start calling it as organal inheritance, because the organelles which are present in the cytoplasm of the mother are actually being transferred or inherited to the offspring. So this we start calling it as cytoplasmic inheritance. So in the later sessions, right, in the next sessions, we'll get into a few examples about the cytoplasmic inheritance, so you get a clear picture what exactly is happening in this cytoplasmic inheritance. Fine.